Good day everyone. Today we're going to discuss about protective coating lipids, the biological waxes. So a biological wax is a lipid that is a monoester of a long-chain fatty acid and a long chain of alcohol. Biological waxes are monoesters, unlike fats and oils which are triesters. The fatty acids found in biological waxes generally are saturated and contain from 14 to 36 carbon atoms. The alcohols found in biological waxes may be saturated or unsaturated and may contain from 16 to 30 carbon atoms. So this means that a biological wax is a type of lipid which is made up of a single fatty acid and a single long chain of alcohol joined by an ester bond, which makes it a monoester. So this differs from a fats and oils which is triesters because they have three fatty acid attracted to glycerol. The fatty acids in the biological waxes are usually saturated and contain 14 to 36 carbon atoms, while in alcohols naman can be saturated or unsaturated and have 16 to 30 carbon atoms. And these long carbon chains give biological waxes their water repellent properties, which makes them useful for protecting surfaces in plants and animals. So the black diagram for a biological wax is, as you can see, the long chain fatty acid and long chain alcohol. So with the fatty acid and alcohol link through an ester linkage, an actual structural formula for a biological wax that we secrete and use as a structural material is this one. However, for waxes, both R and R must belong carbon chains, or usually has 20 to 30 carbon atoms. So in this figure, we can see a biological wax that has a structure with a small weakly polar head and two long non-polar tails. The polarity of the small head is not sufficient to impart any degree of water solubility to the molecule. It means that a biological wax has a molecular structure with a small and weakly polar head and duha ka long non-polar tails. So the head is slightly polar due to the ester linkage connecting to the fatty acid and the alcohol, but it is too small to make the molecule dissolve in water. So the long non-polar hydrocarbon chains of the fatty acid and the alcohol dominate the molecule, making it largely hydrophobic. So as a result, the biological waxes are water repellent, which allows them to function effectively as the protective coatings on the plant leaves, animal fur, and bird feathers. In living organisms, biological waxes have numerous functions, all of which are related directly or indirectly to their water repellent properties. Both humans and animals possess skin glands that secrete biological waxes to protect hair and to keep it pliable and lubricated. With animal fur, waxes impart water repellency to the fur. Birds, particularly the aquatic birds, rely on waxes secreted from preen glands to keep their feathers water repellent. And such wax coatings also help minimize loss of body heat when the bird is in cold water. Many plants, particularly those that grow in arid regions, have leaves that are coated with a thin layer of biological waxes, which serve to prevent excessive evaporation of water and to protect against parasite attack.
Biological waxes find use in pharmaceutical, cosmetics, and polishing industries. Carnauba wax, obtained from a species of Brazilian palm tree, is a particularly hard wax whose uses involve high gloss finishes. Automobile wax, boat wax, floor wax, and shoe wax. Basically, biological waxes are naturally occurring waxes derived from living organisms such as plants and animals. And yes, this wax is used in pharmaceutical. From the word pharma means medicine, and it is used in various of medicinal products. or pharmaceutical. Also, it is used in cosmetics. It incorporated into creams, lotion, and other beauty products. And lastly, polishing industries. It is applied to surfaces to create a shiny finish just like a floor wax. So I know we are familiar in floor wax. We use it to shine the floor. I think it has two colors, the red, white, or maybe I think it has also a yellow one, but I'm not sure. Carnauba wax, obtained from the leaves of the carnauba palm tree, native to Brazil. It is known for being a very hard wax. It is applied in automobile wax, which protects and shines scarf surfaces. Both wax used to protect both holes. And then floor wax provide a glossy finish on floors. And last, the shoe wax polishes and protects the leather shoes. As you can see in the PPT, it has a chemical structure of carnauba wax. So this structure represents an ester, which is a common component of many waxes. So let's proceed to lanolin. A mixture of waxes obtained from sheep wool is used as a base for skin creams and ointments intended to enhance retention of water, which softens the skin. So lanolin is a wax from sheep wool. If you're not familiar of sheep wool is the hair of the sheep. Sheep wool can be used in clothing and apparel. It is used in sweaters, coats, suits, and activewear. Also, it is used in home textile installation gardening like wool blankets, bedding, carpets, rugs, and etc. It is used as a base for skin creams, which provides a moisturizing and protective barrier, and ointments that helps retain water in the skin, keeping it soft and hydrated. A wax is pliable, water-repelling substance used particularly in protecting surfaces and producing polished surfaces. Wax is pliable, meaning wax is easily molded or shaped. In short words, wax is made to protect surfaces and to produce polished surfaces. This broadened definition for waxes includes not only biological waxes but also mineral waxes. Biological waxes is from living organism and mineral waxes is from mineral sources. A mineral wax is a mixture of long-chain alkanes obtained from the processing of petroleum. It is a process of petroleum means a liquid mixture of hydrocarbons that is formed from the remains of marine plants and animals. So mineral waxes, which are also called paraffin waxes, resist moisture and chemicals and have no odor or taste. It is served to use as a waterproof coating for such paper products as milk cartons and wax papers.
So now let's proceed to messenger lipids. So messenger lipids are regulatory lipids that are act in the tissue where they were centralized or the other locations after transport with via the bloodstream. So messenger lipids has its own function. The act of chemical messenger or hormones, the steroid hormones or the sex hormones, for example, the estrogens, androgens, and progestins. We also have the adreneroscorticoids or the mineral glucocorticoids and the glucocorticoids, and also the eicosanoids or the prostaglandins, thromboxanes, and leukotrienes. And now let's proceed to protective coating lipids. The protective coating lipids has also its own function. The first one is to provide waterproof and protective coating. Example, the biological waxes like on leaves, fur, or feathers. They help prevent water loss and add lubrication. So, in conclusion, lipids are essential biomolecules with multiple biological roles. They function in energy storage, structure, communication, and protection. Understanding lipid types helps explain vital cellular processes.
In the next video, we are going to discuss about the saponifiable and the non-saponifiable lipids. The chemical reaction or the saponification. As we all know, saponification, it is a chemical process in which ang fats and oil or the triglyceride reacts with a strong base such as atong potassium hydroxide and the sodium hydroxide to produce soap or the glycerol, which is called the glycerin. There are two types of lipids, the saponifiable and the non-saponifiable lipids. The basis of this classification is a hydrolysis reaction that is carried out in basic solution. What is saponifiable? A saponifiable lipid is a lipid that undergoes hydrolysis in basic solution to yield two or more smaller product molecules. As a result of hydrolysis, saponifiable lipid is broken up into smaller component parts. Bali, ang saponifiable lipid is mo-undergo siya og hydrolysis para makayield og two or more molecules. Ug ang result ani is broken to ang iyang mga molecules kay mo-undergo man siya sa hydrolysis. Next, on the other hand, a non-saponifiable lipid does not undergo na po siya sa hydrolysis and basic solution. Since di man siya mo undergo sa hydrolysis, dili to mo-broke ang iyang mga molecules into a smaller component parts. Ang example ana sa non-saponifiable lipids is the steroids, terpenes, and the prostaglandins. There are 5 types of saponifiable lipids and have 4 types of non-saponifiable lipids. Under the saponifiable lipids is the triglycerols, the glycerophospholipid, the sphingophospholipid, the sphingoglycolipids, and the biological waxes. While the non-saponifiable lipids, we have the cholesterol, the bile acids, the steroid hormones, and the eicosanoids. How could we identify if it is a saponifiable lipids or the non-saponifiable lipids? It is the types of linkage bonds that holds its component parts or the building blocks together. 3 kinds of linkage, all of which have been previously considered, can be hydrolyzed broken by reaction with water. These linkage types are the ester linkage, the amide linkage, and the glycosidic linkage. Ang kanina type sa linkage is ma-found ni siya in the lipids.
Glycerol. So, na siya 3 fatty acids and ester linkage. So, matawag siya triglycerol. So, 3 ester linkage. Glycerol. So, na siya 2 ka fatty acid and phosphate, which is napo siya alcohol, ang phosphate. So, matawag og glycerophospholipid. So, 4 ester linkage. Sphingosine. So, na siya usa ka fatty acids, amide linkage, and na siya monosaccharides, glycoside linkage. So, matawag na siya sphingoglycolipid, 1 amide linkage, and 1 glycosidic linkage. Sphingosine na po siya fatty acids or amide linkage and phosphate alcohol, which is ester linkages. So, matawag siya sphingophospholipid, 2 ester linkages, and 1 amide linkage. Then, na siya ester linkage, which is fatty acid to alcohol. So, matawag siya biological wax, 1 ester linkage. Saponifiable lipids always contain at least one of these 3 types of linkages. The linkage makeup for the 5 kinds of saponifiable lipids is as follows. 1 is triglycerols, so 3 ester linkages. Number 2 is glycerophospholipids, na siya 4 ester linkages. And number 3 is sphingophospholipid, na siya 1 amide and 2 ester linkages.
Saponifiable lipids always contain at least one of these three types of linkage. The linkage make up for the 5 kinds of saponifiable lipids as is follows. First is the triacylglycerols. Second, glycerophospholipids. Third, sphingospholipids. Fourth, sphingoglycolipids. And the last is the biological waxes. The products are simply the building blocks from which each lipid can be considered to have been made. The ester, amide, and glycosidic linkage present are broken through reaction with water, which releases or free ups the lipid's component parts. Any fatty acid molecules released will be converted to fatty acid salts. It's because of the basic conditions associated with the saponifications.
Eicosanoids have structures based on a single fatty acid molecule. Structurally, these non-saponifiable lipids have 2 things in common. First, only one building block is present, and second is there is no need for ester, amide, or glycosidic linkage to be present to link building blocks together, since there is only building one block. So, question: what determines whether or not a lipid is saponifiable? It is the types of linkage or bonds that hold its component parts or building blocks together. 3 types of linkage, of all which have been previously considered, can be hydrolyzed or broken by a reaction with water. These linkage types are ester linkage, amide linkage, and glycosidic linkage. Ang kaning tulo ka buok nga linkage kay makita ni siya sa lipids.